Oh. Can I see it? Can you guys see the screen? Yeah, cool. All right, so we'll start off with immunology. So you guys have a bit of a taster of immunology, and pretty much we always split up different like systems or like barriers or like levels of defense that the body has into three layers: the physical barriers, the innate immune system, and the adaptive immune system. So the smallest one, like probably the easiest one to look at, is the physical barriers, and we have sort of three different subgroups of physical barriers that our body has to defend us from bacteria or like viruses and stuff. So the first one, these physical barriers, which are like, our skin without cracks, obviously, tight junctions between cells, so things can't get through. You've got the cilia and the mucus, pushing stuff up. And even like processes like urinations and things like that, and sneezing. They're like, I guess, processes and mechanisms, but they still act in a physical manner to push things out. So that's the physical element. Then you've got the chemical element, which is like things that will chemically destroy the bacteria or the viruses, like, with different lysosomes and peptides. And so that's like things like the stomach acid and like all the different enzymes we have. They approach the techniques we use to defend chemically. And the final one, it's something quite interesting. Like, you don't think about it, but we have a lot of natural bacteria inside, especially in our like, open areas. So like, our anal canal or like, these like sort of more open areas. It's just lined with bacteria, like the mouth and things like that. And so we call those normal flora because they're bacteria that can live alongside us. We have like this mutualistic relationship with these bacteria. The issue becomes when we use sometimes antibiotics and things like that. They can also kill these things. And that gives rise to things called opportunistic pathogens, which are pathogens that don't normally like hurt us. They're fine. But if you're given the opportunity, if these things die off and they give them space, they'll cause a problem. So that's just that last sort of defense flora, which just take up space and don't let other bad things grow. Then we've got the innate immune system, and this is like the more non-specific approach. And then you've got the adaptive, which is the specific response. So in terms of this system, we look at how like there's a family tree of all the red blood cells, and they'll go through this with you guys. But pretty much we start off with these stem cells in the bone marrow, and they're going to split up into two different sort of pathways: the common myeloid pathway and the common lymphoid pathway. And from that, you can just read the diagram. You've got different things that come off, like different macrophages. So, but what's sort of nice to like a nice way to look at it is most of the non-specific or the innate sort of related white blood cells are from the myeloid pathway, and the more specific ones, generally speaking, are from the lymphoid pathway, which is nice. And then you've got our big boy, the dendritic cell, which is sort of the in-between man, is literally in between. So he comes from his own sort of pluripotent stem cells. So he's not from this pathway or this pathway. He's in the middle, and you'll see it's the dendritic cell counts as both innate and adaptive. It's the bridging sort of cell. Cool. So in terms of recognition, our body uses things called PRRs, pattern recognition receptors, and pretty much the receptors we have that can detect specific things on pathogens that only pathogens have. And these we call PAMPs, pathogen-associated molecular patterns. And so I'm not going to go into big details, but pretty much our immune cells have receptors that can pick up these bacterial specific sort of things. And we have different other ones called DAMPs and other whatnots, but they're not as important. But what is important is there's a specific sort of group of PRRs called toll-like receptors. And these ones they do want you to know because they're more specific, because each one of these, like they're a big family, I guess, and each of these bind to specific bacterial sort of like elements. So you've got TLR3 binds to dsRNA, a specific type of RNA from bacteria, or I think it's viruses. TLR4 binds to lipopolysaccharides, things on bacterial membranes. And then TLR5 binds to the flagellum parts, that wiggly thing that's a bacterium. Cool. So now let's meet the team. We have 5 main cells from this group that we're going to go through. The macrophage, it's pretty much got like all the different roles. It eats, it destroys, it does everything. And it's also a good way to present different like bacteria and tell all other immune cells that, hey, there's an infection going on. Let's do something about it. So that's the macrophage. Just a fun fact about macrophages, when they're in tissues, they're called macrophages, but when they're in the blood, they're called monocytes. So that those two different types of cells are actually the same, but it's depending on what, like the naming conventions, depending on where they are, sort of thing. Then you've got natural killer cells, which destroy infected cells, like with a virus. So from the things that are infected from the inside, or tumor cells, because its job is to kill cells rather than like kill a pathogen. It'll kill our own cells that are bad. Then you've got neutrophils. They're the most we have in the body. They're pretty much the soldiers of the body. And then you've got eosinophils. Their job is to take on things that are too big for us to like phagocytose. So we're going to go through phagocytosis. Pretty much it's a way that our white blood cells deal with bacteria. They eat them and they destroy them from the inside, from like within themselves. But sometimes things like worms and stuff, they're too big. They can't be eaten up. So what these eosinophils do, they just like pretty much, they're really toxic. They just release toxic stuff at these what we call helminths or worms, or these things, and they destroy them like that. So by actually like attacking them from the outside. And finally, we said our Mr. Bridge is the dendritic cell, and it's part of the adaptive immune system as well. And pretty much what it does is it processes these bacterial or remnants of whatnot and presents it outside. Then it goes to the lymph node, and that's where you get the activation of the adaptive immune system that we'll get to later. So now, obviously, as usual, I always have a weekly plug of something I love. So there's a really nice show called Cells at Work. It pretty much details the immune system really well. So have a look at it. I reckon it helps a lot. Like I watched it beforehand and it was really good. So really nice detail in terms of the immunology of the body. Now we'll go to like how actually phagocytosis works. So how does this eating process work? So pretty much what happens is our PRRs interact with the PAMP, then that activates endocytosis. So pretty much the phagocyte goes around and eats it up in a vesicle. Then we get the vesicle that comes around that sort of bacteria is called a phagosome. And that combines with something called a lysosome, which is those organelles we talked about in, I think, our first session, which just have a lot of different enzymes inside of them. And these two fuse together, and so the toxic compounds attack the pathogen, and so you get it being destroyed. And so then the last step is either the cell just gets rid of that stuff, puts out into the extracellular matrix or whatnot, because it just doesn't want it anymore. Or alternatively, if it's a macrophage, it'll just grab it from the inside and chuck it up and display it on what we call MHC2 markers, which are just this receptor that shows an antigen. I'm so sorry, guys. Like I'm going to take a lot of time today. I'll try speeding up, but there's a lot of stuff to get through. That's the process of phagocytosis in image. Hopefully that makes sense. Look at it in your own time. Next up, leukocyte migration. So how is it that our white blood cells get around? Well, they hitchhike pretty much on the blood. They use the circulation to get around. But how is it they get out of the blood? That's the important part, because obviously most of the time our infections are in the tissues, not in the bloodstream. And so to deal with those, they have to leave somehow. And we know that the blood vessels are really they have those tight junctions and any epithelium and stuff. So how is it that these things go through? So there's a process called leukocyte migration, where pretty much neutrophils and whatnot use this process to be able to stick onto the endothelium, roll around until one of their receptors touches a receptor on endothelial cells and they sink through. So we call the two different receptors the selectin and integrin. So selectin is pretty much the rolling thing, and then integrin is what lets it sort of stick and then finally go through. And that last sort of process is called diapetesis. I always can't sort of say it. But pretty much, neutrophils, how they know a sort of pathogens there is that there are cytokines called chemokines, and they sort of make like a trail. So like they're pretty much like dogs, like hunter dogs. They sniff out the smell, they follow that gradient, they go, it's getting stronger, there, they keep going until they get through. And so that's pretty much how you get leukocyte migration. I think there's a question. I'll have a look. Oh no, that's all good, really, thank you. So pretty much, this is a diagram just showing the whole process. If you try searching it up, there's a lot of really complex diagrams out there. Honestly, it's not that important to know the super details, just the general process of selectins, the beginning, and then integrins later on. Cool. Nearly done innate, and we'll get to adaptive. Next up is cytokines. Cytokines are pretty much the comms of the immune system. They're what we use sort of to communicate, all different cells use to communicate. And the main four that they sort of really want you to know are IL-1, which is interleukin-1, and that's what macrophages produce, and it's pretty much causes like all the different inflammation sort of symptoms and activates lymphocytes, which is pretty good. IL-6 is also produced by macrophages, fun fact, and it also does pretty much the same stuff. They're just like really important in terms of activating that whole inflammation, getting everything trapped in one spot so we can deal with it. TNF, tumor necrosis factor, pretty much is something else that's sort of important. You guys generally know it aids leukocytes in their migration, telling them where to go. And then finally, IFN, which is interferons, and pretty much virally infected cells produce it to give a warning to the cells around them that, hey, there's a virus, we have to protect ourselves. That's pretty much it. My goodness. I'm going to do this really brief because there's just a lot. Complement proteins have 3 roles. They're pretty much are these other proteins that help out in our big sort of phagocytomes or our white blood cells. Their main 3 roles are opsonization, which is pretty much the increased likelihood that phagocytosis happens. Lysis, they create pores in the membrane and so kill the bacteria or whatnot. And finally, inflammation, which is they just like get a lot of different immune cells attracted to the area. That's what inflammation does. So that's pretty much what complement proteins do. Read the rest in your own time. A way I remember like the different roles of complement proteins. I always think, hey, a friend's someone who complements you. I had a friend at school called Ollie, but if you don't have a friend called Ollie, you can look at this guy. You guys are probably too young for this show, but it was a really old show. Anyway, the guy's called Ollie. So Ollie stands for opsonization, lysis, inflammation. Thought it was a cool way to just get you guys to memorize that. The last part of the innate immune system that we should probably look at is inflammation. And inflammation is a process whereby we get swelling, we get redness, we get heat, all that stuff. It's pretty much what leads to fevers, which isn't great, I guess. But in the short term, it's super important because it traps the bacteria in one spot because of all the swelling, so they can't get out. And it also gets all these, because there's more like, the redness comes from more blood flow to the area. So pretty much we get more blood flow to the area, more leakage, so more white blood cells get to the area. So we can deal with the pathogen in a closed space. So that's really cool. And yeah, now let's move on to adaptive. So this is the more specific response. And pretty much we also have a team of different cells here. Dendritic cell, again, is the connection. There's CD4 T helper cells, and they're pretty much the ones that aid other cells in activating other like T cells and B cells. Then you've got the CD8 T cytotoxic cells. They're the T cells that kill things. Helper cells, no, they just help out activating everyone else. You know, they stay there. Whereas T cytotoxic, they go out and they kill stuff. They pretty much cause cells to undergo apoptosis. And then you've got B cells, which either become antibody producing cells called plasma cells or memory cells. And we'll get to what memory is later on. So MHC, we brought this up when we talked about the macrophage before in phagocytosis, but MHC stands for major histocompatibility complex. And we have 2, one that all cells have called MHC1, and then one that's specific to immune cells called MHC2. So pretty much what our cells will do with MHC1 is regularly they check out one of the things from inside of them outside and they show it. And that shows that they're a normal cell. But if you think about it, if there's a virus inside, it'll show the virus. And so, or like a viral protein. And so the immune cells will go and kill that thing because they'll know it's infected. MHC2 is a specific type of complex that shows not your own antigens, but things from outside. So bacterial parts or bacterial proteins and whatnot. And that's why it's specific to immune cells. They pretty much eat stuff up and show an MHC2. So how the response starts is our Mr. Dendritic cell, the connector, goes to a lymph node after getting all these different PAPs, like, the pathogen stuff, and they go to a lymph node. And inside the lymph node, all these B and T cells are just waiting there. They're waiting for someone to give them an antigen that they know, that they recognize because it's all specific, and they can activate and they either turn to a plasma cell, make all these antibodies to kill the pathogen, or they turn to a T cell, cytotoxic, to go kill that, thing. But it's specific. That's the most important part that you sort of, I guess, have to take away from today, is that the adaptive response is specific. So there's going to be a specific T and specific B cell for that antigen or for that bacteria. So there are different types of T helper cells. There's 1, 2, and 17. They're the important ones you have to know. And pretty much all you have to sort of get from this is 1 response to viruses, 1 response to helminths, which are like those worms or allergens, and 1 response to bacteria. So that's 1, 2, and 3, 1 to 17 respectively. So how the activation process works is pretty much it's going to be a very big struggle to explain, but help me out if you guys have anything to input, guys. But pretty much T cells have a specific receptor on their surface called a T cell receptor. Really simple, really easy name. But pretty much these helper cells need first the antigen presented to them. And once they see it, they activate. And once they activate, they release all these different chemicals around to get all these specific B and T cells to come in, and they'll activate them as well by showing them that antigen. So we call this process co-stimulation because every cell requires both the antigen and also the T helper cell signal to activate itself. So that's the whole process. B cells are a bit different because with B cells, they actually are allowed to go into the lymph and the blood and circulate around, and they have to pick up their own antigen as well and present that to the T helper to activate. That's something they didn't actually explain in the lectures, and it confused me a lot last year. So I actually emailed privately like the lady, and she was like, "Hey, this is how it works." And so I thought it was pretty cool to just give you guys a heads up before anyone else gets confused like I did, because I'm like, how the hell is it getting this random antigen from? It's a B cell. So yeah, B cells also apparently circulate. So now we'll go to what a B cell actually does, and it's a factory. It makes these proteins that go kill bacteria, and it makes different types, and they'll be different for like what we sort of need. So you got IgM, and that's pretty much the initial response always. Then you've got IgG, which is the late response always. That's pretty much just how to think about it. IgA is something that neutralizes microbes, and it's usually in like a mucus and sort of stuff. Like it's in the secretions rather than like something produced regularly, like or during infection. IgE is pretty much responsible for all the allergies that we have. They're the ones that are sort of not great, but they're also important for eosinophils. And the last one's IgD, which is they assist B cells to find those antigens. Remember we talked about they have to go circulate and find them themselves. So IgD helps with that. Last slide from me. We're nearly done, guys, hopefully. And pretty much it's memory. And it's so cool that our body actually found a way to make us remember different infections we've had. So when they come next time, we have a stronger response to them. And so just when T cells and B cells are activated and they start differentiating to whatever they become later on, some little portions become memory cells, which are cells that stay in the lymph nodes, and they remember that if I'm going to get exposed to this ever again, I'm going to respond like this. So it's pretty much just a faster way that we get a response, because the more we get infected by the same thing, the more memory cells we make, and so the better our response is next time. So that's it from me.
You did. That was a lot. Yeah. Okay. So, yeah. Cool. I'm going to talk about action potentials. So here is a whole big list of very blurry definitions. Just go back and find that image and then save it. So first thing about action potential is the resting membrane potential. So this is when you have just a neuron at rest, not doing anything. The potential is, and what it is it's caused by the difference in the ions between what's inside the neuron, so the intracellular fluid, and what's outside the neuron, so the extracellular fluid. So yeah, so this difference is negative 70 millivolts. So remember that number, please. And then this is called the equilibrium potential. So there is no net movement of charged particles into or out of the cell. There's three key factors which cause this resting membrane potential. So we're saying the fusion of ions down their concentration gradients, but remember at equilibrium potential, there's no gradient. And this is caused by things like mechanically gated channels, voltage gated channels, ligand gated channels, and leaky channels. The next one is the selective permeability. So some ions can move in, some ions can't. And the next thing is the electrical attraction of like the cations and the anions, and that will become important. So chloride. So this is at resting membrane potential. So chloride is principally, it is the anion that is in the extracellular fluid. So this one passively distributes itself. There's no active transport being involved with chloride. And again, the equilibrium potential is negative 70 millivolts. Now, chloride is just a note because it doesn't actually influence the resting potential, but the membrane potential influences chloride. Okay, now we're going to get into action potential. These are some key terms I want you to know and you will need to know. So polarization. So this is the membrane being charged. So that's charged at that negative 70 millivolts. So then depolarization is going from negative 70 millivolts towards 0. So we're getting less negative. And then repolarization is becoming more negative. So towards that negative 70 millivolts. And then hyperpolarization is when it goes beyond the 70 millivolts. So usually, sorry, go back. Okay, sorry. Hyperpolarization is where it goes more negative than -70. Okay, so graded potentials, these are basically, these are the precursors to action potentials. So they're small changes in membrane potential, and they're just local, so at like a certain point on the axon. And they have really short distances, and they can go bidirectionally. These can initiate the action potentials, and they are summative. So the more you have, the more likely you are to cause an action potential. Yeah. So basically, if you have like a dendrite of a neuron, my dendrite, and I have a heap of graded potentials going here, firing into this axon, and I have enough graded potentials, that axon will cause an action potential. So if I have enough to sort of all the way later. Okay, here we go. The threshold potential. So this is an all or nothing. That's key, bold, remember it, don't forget it. So when you have an action potential, you have an all or nothing thing. So if you have enough graded potentials and it polarizes the membrane to -50 or below, to polarizes the membrane to -50 to -55, you are going to get an action potential. So we get the this one here. So we get the depolarization when it gets to the threshold at -55. We get the depolarization, and then that causes the action potential. The depolarization, the repolarization, and the hyperpolarization. And this is caused by the voltage-gated channels. So in each of these phases, you have sodium channels opening and closing and being inactivated, and you have potassium channels being open and closed. So at the rest state, at the resting membrane potential, the sodium channels are closed, but they can be opened. And then once you start depolarizing, the sodium channels are open and they're activated, and the sodium rushes from the extracellular fluid into the cell. And then once you get into the refractory period, these are then inactivated, so they're closed and they can't be opened. And then with potassium channels, you don't have that inactivation, you just have the open and the closed. And so when you get to the repolarizing period, that's when you have your potassium channels opened. That's why it takes longer. It takes longer for potassium channels to open. And then potassium comes out of the cell. And then when they're closed, there's no potassium movement. Okay, so I started talking about the refractory period. So this is like the period of repolarization and hyperpolarization where you can't get another action potential because the sodium channels are inactivated. So you have to repolarize the membrane for it to be for the sodium channels to be in an active state. You also have this thing called a relative refractory period. So the you know how I said before about you need to get like -50 millivolts. Well, in the relative refractory period, you can still get a action potential, but rather than being -50, it might be -30 because that's the threshold is raised. And then in this case, the limiting factor is these voltage-gated channels. So another thing with action potential propagation is because it's unidirectional, it can only go one way because of this refractory period. So if you're going along and this part's in refractory period and this part's not, this part can be depolarized, but this part can't because the sodium channels are inactivated. Yeah, and then the initial site of the action potential will be the first to be reset. Cool. Are you presenting this one?
I think this is lousy synaptic transmission. Yeah, so these are like how two synapses can communicate, or how two neurons can communicate through like little junctions. So we have the electrical, which is through currents, which is essentially direct communication between neurons. These are very fast, and it can go both ways, because essentially think about like little holes between each neuron, just allowing transmission of like the charged ions. And then we have the chemical, which is the release of the neurotransmitters. And if you think about it, that will be slower because you're releasing something into like a little gap between the two neurons. You have to wait for that to diffuse, then for the kind of action to happen. Yeah, and then just other key features just listed there. We kind of the overview, yeah, and then we get also, as we can see here, we have those just like direct communications on the left, and then on the right you have that kind of slow release of those neurotransmitters, and it's also relatively slow, like it's still very fast. And just the next one. So the chemical synapses, so this is like in detail how neurotransmitters are released. So first we have, as Mitch just explained to us, the action potential, and it arrives at the axon terminal, or like the very ends of the neuron that like end up sending messages and communicating with other neurons. So here, that action potential causes the opening of calcium channels. This causes the influx of calcium. And so these calcium that acts on the vesicles within that axon terminal. These then are exocytosed, so that they fuse with the end of the neuron and then are released into the junction. They then act on postsynaptic receptors. And any like neurotransmitters that are in excess, or some of them can even just be after they use, they're just recycled back to that neuron that was just, they were just released from. And then, yeah, exactly. So every time a neurotransmitter is released, it acts on the other neuron, and then this can cause intracellular changes, which then can initiate more propagation of action potentials. Yeah. And then just as we said, so the neurotransmitter will either be recycled, either like degraded, or it'll just diffuse away from the synaptic cleft. Okay, and then just an overview of neurotransmitters. I don't think these are super important yet. Essentially, they can either be excitatory or inhibitory. I personally don't think you need to learn these stuff, but there's just some information here. These are just examples. Yeah. So excitatory makes sense. You're going to cause a depolarization, and inhibitory, it's going to cause it to be more negative, so hyperpolarize it. And that's going to make it harder to reach that threshold to then generate an action potential. And obviously a lot of graded potentials or lots of excitatory neuron kind of signals will end up causing eventually an action potential. Yeah, next please. Okay, very basic receptor generic vibe at the moment, which you'll kind of learn a bit more later on. But so on the left, we have our ionotropic receptors. These are just essentially a receptor where one ligand will bind to it, and this causes just one action. So it might just open the channel, things go through. Then we have the metabotropic, I don't know how to say that, receptor, but something will act on it, and then there'll be changes within that receptor, which then slowly trigger other events. But there's also secondary messengers from that receptor coming, and this also allows for an amplified response. So even though it's slower, it's going to amplify. So think of ionotropic as direct, and the other one is not as direct, but causing a larger kind of like secondary causes and interactions. Cascading effect is the word we use there. Yeah, that's about all from there. And this is, I think, a perfect diagram to show. So on the left, we see how it just goes straight, like the ligand, the neurotransmitter or ligand or whatever, binds to the channel, allows just direct flow. But then on the second image, we have that neurotransmitter binding to the receptor, then causing release of those G proteins at the bottom, which then might add to something else, change something inside the cell, which then activates maybe another ion channel to open and allow those flow of ions. So yeah, that's that. And now we have our vital signs.
Okay, hello everyone. So basically with vital signs, you'll go into a lot more detail of it as you go through year 1 and 2. We never did vital signs in year 1 because we were online, so they decided to wait till year 2. So you might not cover all of this in like 1 week, but it's kind of good to know, and a lot of it makes like logical sense in things like temperature. I'm sure you've all had your temperature checked once in your life, so it's something that kind of makes common sense as well. Okay, so yeah, if we look at temperature first, basically there are 4 main ways that we check temperature. Mostly with all the vital signs at the stage you're at now, the most important thing to kind of know is like what's considered normal, because obviously you're not really going to be checking people's temperatures or blood pressures till you kind of go in like clean years. For oral, we obviously have this range here. Just to be honest, they have never asked us this range as of yet, but it might be important to know. What is probably important to know is that the eardrum and the rectal both are reflecting the core body temperature, whereas the others, as you can see, are all a little bit different. So the oral is a little bit lower than the eardrum and the rectal. You'd obviously also never really be expected as a student to check the rectal temperature of a young child at this stage. So it's important to know, but you probably won't be doing that. Okay. Next, we have the pulse rate. So again, this is important. It's obviously really important to know what the normal resting heartbeat is. And what you'll find as well is like when you do more micro and look at infections, they'll give you cases where like they tell you what the patient's heart rate is. And so it's important for you to be able to know whether it's normal or whether it's high or low. Usually we check the pulse rate at the radial pulse, which is on your wrist. And what you do is you put, I feel like most of you would have seen it or like in medical shows and stuff, but basically you just place, they say you can place 2 or 3 fingers over your wrist here where your radial artery is. Then you wait until you can feel it. And basically, if it's considered regular, so like if you can feel that it's like beating at a normal rhythm, then what you can do is you can count it for 30 seconds and multiply it by 2. If you feel it and it's a bit irregular, like it's not beating at a consecutive space apart, then you would want to count for 60 seconds, and that would give you your beats per minute rate. Okay, then rest rate. So again, the normal range is 12 to 20, and it's important to remember this is only in adults, so it can change in children, but you don't really look at children as much at the moment. What's really, really important with the rest rate is that the patient shouldn't know that you're checking their rest rate, because as soon as someone knows, like if you know that someone's watching you breathe, your breathing pattern will change. And so what they tell you to do is they tell you to like check their pulse and pretend that you're still checking their pulse. But actually what we do is we watch their chest rise and fall. And each of those is obviously going to be like 1 breath. For rest rate, because they don't breathe a lot in like a minute, you want to count for 60 seconds straight. So you don't want to do the 30 seconds and times by 2. It can also be really hard where we were practicing this in clin skills, and it's really hard sometimes because you've just measured their pulse, and so you're trying to remember the number of the pulse while counting the rest rate. I think it would probably just take practice to like get good at both. It's also sometimes really hard to see their chest rise and fall, like if they're wearing a jumper. Our clin skills tutor said like change. Next we have pulse oximetry, which is the easiest one. Basically, I found it really interesting to learn how it worked, is that you might have seen in like hospital shows and things like that. It's where they put the little clip on your finger and it gives a reading of your oxygenation level. And basically, the oxyhemoglobin and deoxyhemoglobin absorb the infrared light differently, right? So what it does is it projects infrared light and it measures how much is absorbed. And by that, it can tell the like percentage of deoxyhemoglobin you have and the percentage of oxyhemoglobin you have. The normal range is 95 to 100, but if it's like less than 98-ish, you should probably like check it again. Like 95 is okay, they're not dying, but like it's not ideal. So really the oxygenation rate should be as close to 100 as possible. And I just thought I'd that's how it's sometimes written, SpO2, which means saturation of peripheral oxygen. So if you see it written as SpO2, that's what it stands for. Okay, and the last one is blood pressure, and blood pressure is probably the most confusing one to do, and it definitely will click more when you get to practice it in person. Again, we have a normal blood pressure of 120/80, and that's saying that's your systolic blood pressure over your diastolic. Systolic is when your heart is pumping, like the contraction, and the diastolic is when it's relaxed or when it's chilling. This is just a little table that I found in the notes where this is like different ranges. So it's good to know like where they're falling into, if that makes sense. So, okay, if we go to the next one, Mitch. Next slide. Thank you. Okay, so there's 2 steps that we have to take when we are measuring blood pressure. Basically, I'm sure you would have all seen like the pressure cuffs before. And so I'll show you on my hand, basically where you put it and stuff, because it can get kind of confusing. So what you want to do is you put the blood pressure cuff, as you probably would have seen, like here, like around your biceps, just a little bit above your little arm crest. I don't know what you want to call it, like where your elbow is. So what you want to do is you have the blood pressure cuff on and you want to squeeze it. And by the way, you also need a stethoscope for this, which is why when you actually get to practice the clin skills, it'll make a lot more sense. In saying that, when we had our clin skill session on this, no one could do it. Like you just are waiting to hear these sounds and you just never hear them. So it can be really tricky. But what you want to do is you want to, when usually, if you put a stethoscope over your brachial artery, which is here, you will hear a sound, like because the blood is flowing through it. And so what you do is you put the pressure cuff on and you pump it all the way until you can no longer hear a sound. And what you do then is you really slowly want to start releasing the pressure until you hear a sound again going through the stethoscope. And the point at which you can hear a sound again is the point at which you have your systolic blood pressure, because that's the point at which blood can start flowing through again, right? The heart is pumping hard enough that it can it matches up with the pressure of the cuff and therefore can start flowing through the arm again. Then you would record that down. So that's just to find your original systolic pressure. The next slide, we'll talk about the main part of checking blood pressure. And thank you. So that's called the I can never pronounce this one, Auscultatory method. I don't know how to say it, method. So what we do, again, we have our blood pressure cuff at the same place and our stethoscope over the brachial artery. It can be pretty tricky to find where the brachial artery is, especially when you have the blood pressure cuff on. So that just kind of takes some practice. Okay, so what we do is we've already recorded our systolic blood pressure. And now what we want to do, and this can be really uncomfortable for the patient, so you don't want to do it for too long because you're going to pump it 30 milligrams of mercury above whatever the value you got was. So if it was 120, you would pump it to 150. So it's pretty tight on their hand. So what you do now is the first part is pretty similar to what we've just done, but again, you're going to slowly release the blood pressure cuff until you hear a sound, and that's going to be your systolic blood pressure. Then, as you keep releasing, the point at which you can no longer hear a sound is called your diastolic pressure, and that's because that doesn't mean that there's no blood flowing through the veins and arteries, but what it's saying is you've got such a even flow going through that you can no longer hear a sound, because usually you can only hear sounds when there's like muffles or murmurs, because it's like once it's an even flow, it just goes through. It's like water going through a pipe. So the most important things to remember is the first sound you hear is when the blood starts to trickle through again. So that's going to be your systolic. And then the last, when you don't hear any sound anymore, that's going to be your diastolic. obviously nowadays people use like the mechanic, like the automatic cuffs, the ones that just measure it yourself. But obviously in things like OSCEs and stuff, you're going to have to do it by hand. And when you practice it, it does make a lot more sense. However, it is really hard to find the brachial artery and hear the sounds. I don't know if anyone else struggled, or maybe it was just my tube group, but it was pretty tricky. And yeah, those are the 5 vital signs.
Yeah, and then, sorry, I have the spinal cord. I found the spinal cord very confusing initially because I think they just initially describe what's happening and you're like, I have no clue of the anatomy. I don't know any of these words they're saying. So I think this is good just to like maybe try watch this or kind of understand this before you read. You get to, I think it's called like ascending, descending tracts lecture. All right. So just like it's probably important to know like what the actual spinal cord is. So essentially all your nerves in your body have to like communicate to the center, and they travel through the spinal cord or they come off the spinal cord. It has motor fibers, sensory fibers, fibers that you can't control. So it's your like autonomic nervous system. It's in charge of your reflexes, and yeah, just connects your brain to your body. And it's important to know that there's 2 pathways. So we have our afferent sensories. These always go towards the brain because you're receiving this input. And then we have the efferent motor. These goes away from your brain, so it goes down, out, and that allows you to implement action. Yeah, and then to the next one, please. Sorry, the formatting really changed here when I switched the slides over. I'll fix that up before. But the key thing to know is same day, which means sensory is your afferent, motor is efferent, and dorsal is afferent, ventral efferent. So remember, afferent and sensory, same thing. They're going towards your brain. Efferent means out, motor, they're going away from your brain. Dorsal also means posterior, so the back. Imagine like a dorsal fin. Yeah, that's how I always remember it. And ventral is the front. I'll touch more on this on the next slide, but all your sensory is going to come through the back part of the spinal cord up to the front, and then all your ventral, all your motor is going to come from the front, out, come down from your brain, and then out through the front. So I don't have much, you can't see my mouse, but what, yeah, so we have the dorsal at the back, ventral at the front. Very important to always orientate yourself. Then we have these mixed spinal nerves, which come into the spinal cord. Yeah, and then they're going to separate out. A mixed spinal nerve means it has both your sensory neurons, which are at the back, and your motor neurons, which are at the front. But I'm going to go into a better diagram on the next page. Another important thing to note is this: what the actual cross section of the spinal cord shows is this white matter and the grey matter. Imagine grey matter, all the cell bodies, the ganglions, the dendrites, and then the white matter is kind of the fatty buildup of the axon myelin sheath that goes up and down, which is what causes that white matter of the spinal cord. So you can think about all the whites are going vertically up and down, and dorsal is more horizontal movement. Yeah, and we can go to the next one. Ibi, you might have to help me out just with the, so I'm just going to do very basic what's happening. So everything here is labeled. I'll just go through first. I'll go through it twice also within the terminology. So let's say we have something like you touch something. So we have the skin, you have the little X, the V at the bottom, you're saying it's receiving information from your skin. It then goes back through this red, yep. And then it's going to have, this neuron has its cell body in the dorsal root ganglion, which they sometimes abbreviate to DRG. It's the same neuron, which then continues through the dorsal root, and then synapses onto another neuron, or might go up and down on the dorsal horn. And the green one in the middle is just an interneuron for when there's a reflex. But this can synapse to another one, which just goes up and down, or it itself can go up and down and synapse somewhere else. But it will pretty much synapse on that spinal cord level, almost always. Then we have the ventral. So let's say, I touch a table and I'm like, that doesn't feel so nice. I'm going to touch something else. So I'm saying to my brain, I want to move my hand. Brain, whatever, you'll learn a lot about motor, like coordination later in Sem 2. Your brain sends signals down. It's then going to go from the ventral horn. This is one motor neuron coming out. So there would be another one telling it to do this, but it comes out down the ventral horn. From the ventral horn is the cell body. Then it comes down, the axon goes down ventral root, through the mixed spinal nerve, then out to the skeletal muscle. And that's kind of the very basic setup of how this is arranged. What's important to know is nerves go in to the spinal cord initially mixed. So we have then, let's say this is sensory, this is motor mixed, and then they split. We have one going to the back, the dorsal, which is always sensory, remember same Dave. And then we have our ventral, which is the motor, that's also remember same Dave, which then goes to the front. So sensory is always at the back, going through the back path, going into the spinal cord through the back, and ventral's always the motor nerves coming out of the spinal nerves through the front. Lots of this will make a lot more sense as you work through lectures and kind of things like that, but hopefully this is a good overview. Important to know as well is that the green kind of neuron I put in the middle is called an interneuron. And this is if you need something like a reflex. So your brain or your body has a mechanism whereby reflexes are made so that you don't have to go all the way. Let's say you touch something hot, you don't want to have to go all the way back to your brain, send a signal, then go all the way back down, send out. So instead, your skin touches something hot, it goes straight to your spinal cord in the middle on that gray matter. It directly synapses onto a interneuron, which is that green one, which then directly synapses onto the motor neuron out. This is called a polysynaptic neuron because there's 2 points of, or more than 1 point of synapse, but it's possible for the sensory neuron to synapse directly onto your skeletal muscle neuron and then have that direct, that's called a monosynaptic reflex. So it doesn't have an interneuron between it. Awesome. I think that's about it for the generic kind of basic spinal cord arrangement. It does get a tiny bit more complicated when you add like autonomic nerves in, but we're not going to worry about that for a while. Keep coming back to this image and just remember that acronym same Dave. I think I have one more slide, or maybe that's the end. No, that's it. But I really recommend reading, like going through this, because I think you would have the same lecturers who we had last year, and he does not explain this well, I don't think. Michael, there is a workshop which makes it a lot more a lot easier to understand, but the workshop is not situated before the lecture, so it makes it hard to like understand that concept until after. But yeah, that's about it. Did anyone have any questions?
Just like highlight is, Lily said this really well. She said that the body of the sensory one was in that dorsal root ganglion, whereas the body for the efferent ones, like the one that does something, is in the spinal cord. That's something really they always test you on. They always ask you questions about it. So just remember that one's in that dorsal root ganglion, special DRG place, and one's in the middle of that spinal cord.
Which is why that diagram that I showed at the end, I think I actually maybe got that directly from the lecture. It's actually just like a really simple diagram that actually has a lot of high yield information in it.
Cool. Any questions?
If not, thanks for coming. We did go really long today, but it was because you have a lot of content. Enjoy that. You should have enjoyed that mid-sem break because you needed it before this week. But it's fine. It's manageable, especially if you like take it slowly. Like if it doesn't make the sense the first week, it's fine because they keep coming back to these concepts. They're things that are going to stick with us like forever. And so pretty much the more you get exposed, the more you understand, the better it's going to be. So like, don't worry about it. But I remember this week was very difficult, but once you get through it, it's fine. So yeah.
This is also one of those things that when you return to it, it's a lot easier to understand. It's just like one of those things that just clicks and you don't even realize it. And then you come back and you're like, what was I struggling with? But it's really overwhelming at the start. And also just like, there's a lot of information to take in, especially with immunology. And if you didn't do bio, which I know I didn't, so I felt I had a lot to kind of catch up with there. So I found like making my own diagrams of what's happening really helpful, because there's a lot of cell interactions and things you can draw out and like processes like that. Yeah.
Thanks for coming, guys. And we'll hopefully put this upload soon, and we'll upload the slides as well soon. So stay tuned for that. Thanks for coming this week, guys, and hope you have a
Thank you
great
so
week.
much. Thank you.
Thank you, guys. See you.
Thanks.